Hi, fellow Genetic Engineering Solutions Group. Kyle Holland here with you, and I'm happy to present on enteroviruses today. Start with a quote. The single biggest threat to man's continued dominance on the planet is the virus. This is from Joshua Lederberg. I think this rings true today. I took some editorial liberties to update it to current times. And I think the development of genetic engineering and sequencing solutions for viruses will allow science and technology to advance in combating disease-causing pathogens. Enterovirus is a genus of viruses in the Picornaviridae family, and the family of viruses are a group of related non-enveloped RNA viruses which infect vertebrates, including mammals and birds. The root word entero is Greek in origin, and it means gut or intestine. Many of the enteroviruses achieve transmission through the mouth, and they are hardy enough to survive the acidic pH environment of the stomach and pass into the intestines. From the intestines, enteroviruses can enter the bloodstream to travel and spread infection in other areas of the body. So what is a virus? We know a virus is a small collection of genetic code, either DNA or RNA, surrounded by a protein coat. We know viruses need host cells to reproduce. A virus cannot replicate alone outside another cell. Therefore, it is not considered a living organism. We know viruses are different than bacteria. Each have different components. Viruses cannot be eradicated by antibiotics like bacteria can. Only antiviral medications or vaccines can eliminate or reduce the severity of viral diseases. This includes AIDS, COVID-19, measles, smallpox, and hopefully soon enteroviruses and all viruses. RNA viruses have genomes made of ribonucleic acid, RNA, and can be separated into three groups. The group that we are going to focus on in this presentation is positive sense single-stranded RNA viruses. So we'll see this plus ssRNA a few more times throughout, and that's what it stands for. This is David Baltimore. He is a virologist and biologist, professor right now at Caltech, and he discovered reverse transcriptase. The reason he's included here is because he has come up with a virus classification system named after, it was named after himself. And class 4 includes the positive sense single-strand RNA viruses, which enterovirus would be classified under. So the positive sense single-strand RNA genome works as mRNA. We don't need transcription required for translation. So this means enteroviruses are able to make more viruses in fewer steps, which makes them difficult to combat, and they can quickly take hold in a host and cause a lengthy list of problems that we will discuss a little bit later on. This gives you kind of an idea of how viruses are separated, classified. We'll start here with the RNA virus, and then it is single-stranded, and it's positive sense, and it is naked, meaning non-enveloped. And this is our family of viruses that enteroviruses fit under as a genus. This is the shape of enteroviruses, most of them, and it's a common shape, icosahedral. We have the geometric triangles that are all linked together that kind of look spherical, but yeah, they're not exactly round. This is the outer capsid layer protein, and inside would be the positive sense single-strand RNA virus genetic material on the inside. Here, this is kind of a map that we can follow. Positive sense RNA viral genome here, except while it can make negative sense single-strand RNA, not DNA, and not the DNA polymerase, like we learned in a previous module, it would be RNA-dependent RNA polymerase. So we can skip a lot of these intermediate steps, and the positive sense single-strand RNA would essentially be mRNA that can go straight to translation for viral proteins. And it can produce a lot of these viral proteins to form a capsid where the genetic material would be housed inside. The positive sense single-stranded RNA is the template for negative sense single-strand RNA that can produce many more positive sense single-stranded RNA with RNA-dependent RNA polymerase. So basically, these viruses can put their genetic material into a cell and make more viruses without all of these intermediate steps, which makes them very efficient at replicating and spreading throughout the body. Enterovirus transmission is typically gone through the fecal-oral route, and it's oftentimes hand-to-mouth contact. Washing hands is important. I think over the past 9 months, the global population has learned the value to frequent hand washing, and this has helped reduce the introduction of pathogens by way of hand to mucous membrane contact. And once a person is infected with enteroviruses, they can spread person to person from direct or indirect contact. Here are some of the exposures that people can come in contact with to enteroviruses. Feces, obviously, if you're working in healthcare facilities where you're having to change people's bedpans or diapers or things like that, it's obviously very important to wear the proper protective equipment and make sure that the hand washing and cleaning of surfaces is trying to reduce the risk of exposure there. Information from the CDC here: enterovirus infections in the US that produce symptoms in people each year between 10 and 15 million. This gets reported annually by the CDC. And enteroviruses are worldwide, so throughout the globe. They have an interesting seasonality that we will get into. They prefer hotter temperatures, so it's a summertime virus. The peak months for infectious activity are June to September in the Northern Hemisphere, and then it would be the opposite in the Southern Hemisphere, so December through February. And the Japan World Study on hand-foot-mouth disease, which is an enterovirus, shows a trend of every other year an increase in cases. And we'll see that here in this. We have case numbers here. On the y-axis, we have weeks here on the x-axis, and I like this colorful bar here: June, July, August in yellow, and then September here in the pink. And these are our most active months. And if you follow the even months, the even years, 2012, 2014, 2016, 2018, they're all relatively flat. And 2013, 2015, 2017, 2019, the odd years all have this rising case activity. So the seasonality of it for the Northern Hemisphere is going to be in the summer months, and they are more active in the odd years than the even years, shown by this chart for this particular enterovirus that causes hand, foot, and mouth disease. Our highest point here is right around 43,000 patients or cases, and this looks to be at about week 30 or 31 in the year of 2019, so July. What this trend would lead me to hypothesize would be for the summer of 2021, you would expect to have a big increase in numbers again following this trend. However, with the amount of hand washing and mask wearing throughout the world, I somewhat anticipate the numbers to decrease over the next summer 2021. Here are some of the symptoms seen by the more mild enterovirus infections. These are fairly insignificant and oftentimes don't require any assistance from medical professionals. Here are some of the more serious infections and damage they can cause to the human body. In one of the case studies that we'll be looking at, you'll see the viral meningitis is incorporated, as well as the infection and swelling of the sac around the heart, and the viral encephalitis as well. Enteroviruses are diagnosed through lab tests, and they recognize them using polymerase chain reaction, as well as the enzyme-linked immunosorbent assays. Currently, there are no treatments available for enteroviruses. For the more serious ones, like Enterovirus 71, it's notable for its role in causing epidemics of severe neurological disease and hand, foot, and mouth disease in children. Antiviral therapies may be utilized in the case of EV71. But most oftentimes, infectious disease doctors build personal care plans for the individuals to manage symptoms. The doctors we just spoke about are encouraged by the recent advancements on RNA vaccines. The SARS-CoV-2 is an enveloped, positive sense, single-stranded RNA virus, as are most enteroviruses, positive sense, single-stranded RNA viruses. And the advancements made on the vaccine for COVID-19 shows a lot of promise and potential in aiding the eradication of other diseases caused by enterovirus pathogens. This is the case study of this 33-year-old woman that did have pre-existing, she was immunocompromised and was infected with Coxsackie virus B type 4, which is an enterovirus, and she succumbed to the infection. And what I found most interesting about this case study was the blood work that we see here. This is actually a cerebrospinal fluid analysis, and the neutrophils here, as part of the innate immune response to the invading organism, we follow These are the days of hospitalization here from day 4 to day 6 to day 17. And this is the differential count, so the percentage here, 86 neutrophils to 14% lymphocytes. And we see the numbers slowly changing here. And then this is where the lymphocytes take over as part of the adaptive immune response, and the neutrophils are less involved. And unfortunately, this case study, the woman succumbed to the infection and did not make it, but the blood work, or the cerebrospinal fluid analysis, I thought was interesting to recognize between day 6 and day 17 the change from the adaptive immune response from the innate immune response. These are the references that I used. I want to thank them for all their hard work. And I also want to give credit and a special thanks to all the people at Slides Carnival that made this presentation free and available for students and professionals to openly use. I think it spiced it up a little bit. Thanks for watching. Hopefully this one was shorter than the last one. And if you have any questions, please email me, Kyle_Holland@elcamino.edu.